Good morning, students. The next topic that you have to study is natural polymers. We just need to study it briefly. So, natural polymers are those polymers which exist in nature. Examples could be your polysaccharides, peptides, proteins. Your DNA is also a form of polymer. So all these are natural polymers. Other than these, there is natural silk, wool, cotton. All those also come into the natural polymer category. So polysaccharides are basically polymers of monosaccharides. And peptides and proteins are synthesized from polymerization of amino acids. So if you have to synthesize polysaccharides, or you can say sugar, you synthesize it from monosaccharides, or you can say you can synthesize simple sugars. You can also convert these simple sugars to carbohydrate. So what are monosaccharides? Monosaccharides are aldehyde and ketone with 5 or 6 carbon atoms and many hydroxy group. They are simple sugars that are the monomers of carbohydrates. Two most common sugars, D-glucose, D-fructose, are given here. In glucose, we have this aldehyde, and in fructose, we have this ketonic group. Other than these two functional groups, there are a lot of hydroxy groups present here. And these sugar not only exist in form of straight chain compound, but they also exist in the ring form. So they can exist both in the linear and ring form. If you look at the structure of glucose in the ring form, this is the structure, or more accurately this one. Now how these monosaccharides are converted into polysaccharides. Glycosidic bond formation takes place between two monosaccharides upon condensation reaction. We have studied two types of polymerization. One is addition polymerization, another is condensation polymerization. So in case of saccharides, two monosaccharides undergo condensation reaction, resulting in the ether linkage. What happens? Two hydroxy groups of any two monosaccharide react. Which two hydroxy group will react? It will depend upon the disaccharide being formed. So if you look at this, in this page, sucrose has been given. 1 molecule of glucose, 1 molecule of fructose undergo condensation reaction. A water molecule is released, and ether linkage, or we also called it as glycosidic bond. Glycosidic bond formation takes place, and this molecule obtained on combining glucose and fructose is sucrose. Similarly, Lactose is a sugar which is found in milk. So lactose is a disaccharide actually, because it has two monosaccharide has combined to form this molecule. So if you look here, this hydroxy of galactose and this hydroxy group of glucose undergo condensation reaction. One water molecule is released and one oxygen forms this glycosidic linkage or this ether bond, and lactose is being formed. So sucrose and lactose we have seen. Similarly, if we have some repeating units or some different type of monosaccharides, depending upon that we will obtain different polysaccharides. If you look at cellulose is synthesized one for glycosidic linkage. Similarly, simple starch, this also have one for glycosidic linkage, but when you look at glycogen, it has two type of linkages, one for glycosidic linkage and 1,6 glycosidic linkage. So in this way many different polysaccharides are synthesized. If you look at cellulose, starch, glycogen, chitin, all of these are example of these. Next is amino acids is a monomer for peptides and proteins. There are various amino acids that exist in nature. Amino acid is an organic molecule which have two functional group, carboxylic acid and amino group, and a hydrogen atom present in all the amino acids, and the fourth group will depend upon which type of amino acid is there. So if you look at the journal structure of amino acids, this is carboxylic acid functional group, this is amino group, and this is hydrogen group. And for simplification, R is written for the fourth additional group which is present in amino acids. It could be written like this or written like this. If we vary our R group, different amino acids are obtained. Let's look here. These are aromatic groups. If we have R as CH2 and one benzene ring, then this becomes phenylalanine. If this one have one hydroxy group at para position, then it become tyrosine. So this way we name different amino acid. If you look at the simplest, this is glycine, where R group is a hydrogen. If R group is a methyl group, it is alanine. If it is propyl group and attached from the second carbon, then it is valine. So that way there are different kinds of amino acids. Those could be polar, those amino acids could be non-polar, positively charged, negatively charged, or your aromatic. Some 20 amino acids exist in nature, and these amino acids are chiral compounds. if we leave glycine, all the other amino acids have 4 different groups present on this carbon. Due to that, this central carbon becomes chiral. So the amino acids exist in 2 enantiomeric form. One will be rotating the light to the right hand side, plane polarized light to the right hand side, other one will rotate it to the left hand side. So basically amino acids are divided, the enantiomers are written as L enantiomer and R enantiomer. Naturally only L amino acid exists in nature. So this is what I was saying. Chiral carbon, this carbon is chiral. They have taken up the example of alanine. So D-alanine and L-alanine, these are 2 enantiomeric forms. Only L enantiomer exists in our body. Now how the proteins are synthesized from amino acids. So again there is a condensation reaction happening, and in this time when 2 amino acids combine, carboxylic acid group of one amino acid and amine group of the second amino acid, both of these combine, undergo condensation reaction, a water molecule is removed and a amide bond is formed. This bond in case of amino acids is called peptide bond. And when 2 amino acids combine, it is called as dipeptide. So amino acids have peptide bonds. Now when we have a straight chain of very large number of amino acids, it is called primary structure of the amino acid. Like here you could see the example. So here we have 4 different amino acids present. So it's a sequence of 4 amino acids shown here. If you have this kind of chain, it is the primary structure of proteins. Now we have 20 amino acids, and there are lot of permutation combination possible. We can arrange 20 amino acids in an infinite number of possible sequence. So various large number of protein structure can exist. Now you have to identify which amino acids are present in this polypeptide. Here previously there was a page in which all the amino acids which exist in human were shown. You can refer to that and name the amino acid present in this sequence, and using that only draw a tripeptide of tyrosine, aspartate and lysine. Other than primary structure of amino peptides, proteins can exist in primary structure, secondary, tertiary, quaternary. Basically proteins exist in this way, but there are different structures that we name. Primary structure is a straight chain sequence of amino acids. Secondary structures are obtained when they attain some kind of two-dimensional structure due to the presence of various interacting bonds. Those bonds could be your hydrogen bonds or any other non-covalent bond like ionic interaction, ion dipole interaction. So due to the presence of non-covalent interaction, these primary structures get converted into a two-dimensional structure, or I should say three-dimensional structure would be correct. They could exist in a helical form or a pleated shaped form. Now, on further arrangement, tertiary and quaternary structures are obtained. So our primary purpose here was how the proteins were obtained from amino acid. So the amino acid undergo condensation reaction and form peptide bonds, and with each peptide bond formation there is a release of one water molecule. So that was the sole purpose of reading it here.